Let's go. I shaved a little bit. Anyways, let's put on a shirt though and clean up. Test, test, test. Test, test, test. Testing 1, 2, 3. Oh, yeah, I think. Hello. Nemo. Dora. Hello. Maybe it doesn't work. Hello. It's working just fine. Hello. Test, test, test. Test, test, test. Oh, maybe right there. Or somewhere between right there. Hello, hello, hello. Maybe right there's better. Anyways, look at that. Balding. I'm messing. I don't really care. Okay. Okay, let me get some bread, guys. No more. Anyways. Alright, we're back now for good, so we're fine. I think you're just gonna rest today. Maybe study more. I don't even need a fan anymore. Stuff right there. I can tell which one's the 16-minute timer and the break timer. Because the break timer button is more pushed in. I think the spring is weaker. Let's clean the desk before we start. Oh, hey Nadine. Yeah, I got them from Amazon. Maybe I can put the link up. Did you want to buy them? Looking to buy them? Oh, okay. Yeah. Maybe I can just put them up. Put up the links. Maybe I can put in like. Well, I'll just put it here for now. Hold on. Let me see if I can find it. I found it right here actually. I don't know. Oh, I'm not. Yeah, that's fine. Yeah. Okay. I'll do it later. Okay, let me put the food downstairs, and then I'm gonna clean the desk. Is the audio okay? I mean, it seems like the audio is okay today, right? The microphone. Yeah, cool. Very nice. Thank you. Okay, let me put the food downstairs. I'm gonna clean up, and then we'll get started. Where's my headband? It's a little bit too hot. Okay. What's the good rules? Hot stable, redundancy, and the data code. Hold the sister components together, and is then dissolved after anaphase during mitosis. Is it dissolved? I guess it's dissolved. Maybe it's dissolved. I'm not sure. I think so. Splicing, 5' cap, poly A tail. What is that noise? Yeah, splicing. Allows any placenta glutamate allows any placenta neuron and this action potential or change in voltage, right? Oh, okay. Well, depolarizes. Secondary structure. Side chain interactions. Secondary structures of hydrogen bonds. Histones, H2A, H2B, H3H4. And then and then that's stuck. Is it stuck together, I guess, by H1? DNA. Oh, that's how the DNA actually wraps around. It's because of the H1 histone. H1 black stick holder. I don't know what you call it. holder. Okay. So any plus intron, and this depolarizes peptide bonds. The region of the brain, the thalamus, hypothalamus, and the basal ganglia. This is the hindbrain. This is the hindbrain. Wait, what? Forebrain? Oh, whoops. Wait, I thought that was a hindbrain. Oh man.
17 different structures in the brain, kind of lay out.
Let's just watch a quick video.
The basic plan of the brain, but you're using your brain to process it. And if you do a good job when we get to the end and I review all the parts, you should be able to tell me what their structure is and what their function is. And so what type of organisms have brains? It's the animals. Animals use nerves, they have muscles to move around, and so they have to organize that movement. And so they use a brain. And so if we look at the two basic body plans of animals, some are radially symmetrical. In other words, they're built around almost a tire. And then some are bilaterally symmetrical. In other words, a tiger, you could draw a line right down the middle. There's going to be a clear right side and a left side. There's going to be a clear front and end. And as we became bilaterally symmetrical, we had to organize that movement. So this is a simple animal body plan. And so this animal is going to move towards the right. And as it does so, it has to take in information. We call that sensory information using neurons. And so right now you're taking in sensory information from your eyes, from your ears, and then inside your brain, you're going to integrate that information. You're going to make sense of it. And then you're going to figure out what you want to do, how you're going to act dependent upon that. And so then we have this loop of motor neurons out, or motor nerve. And so this loop in simple animals is also important in understanding how our brain works. But if we look at these real primitive brain, we find that they have a real common structure. They have these 4 humps, and we call those the first one's not a hump, but the spinal cord. We then have the hindbrain, the midbrain, and then we finally have the forebrain. And we find this consistent throughout all animals.
I have to finish this paper.
It pretty much looks just like that primitive brain. You can see down here, we've got the spinal cord that's bringing information in. We then have the hindbrain, the midbrain, and the forebrain. And so one thing you should remember is that the closer we are to that spinal cord, the more basic the functions are. And so right down in this hindbrain, it's going to be basically keeping the heart beating, keep the circulation going, digestion in the shark. But when a shark decides to attack you or it has some kind of an emotional response, that's going to be way up here in the forebrain. Now, if we look at you when you were really little, when you were an embryo, you had a brain that looked very similar. You had a spinal cord, you then had a hindbrain, you had a midbrain, and then you had a forebrain. But during development, that brain changes radically. And so this is what an adult brain looks like. So we still see that spinal cord. We then have the hindbrain, we have the midbrain, but look how large that forebrain is going to be. And so that's where all of those emotions and memories and all of that thinking we generally attribute to the brain is going to be the forebrain. And so let's get to the actual anatomy. So there are going to be 17 parts that we're going to go through. So you should always be thinking, what's the name of the structure? Where is it? And then what's the function? What does it do? So if we look at a basic brain plan, we find these 4 things jump out right away. We're going to see the brain stem. We then see a cerebellum on the back of the brain. So again, to get yourself oriented right, the eyes are going to be right up here. And so this will be towards the back of the head. So that's going to be the cerebellum. We then have the area of the thalamus, hypothalamus, and then finally we have the cerebrum, which is going to be that dominant upper portion of the brain. And so let's begin with the brain stem. The brain stem is broken down into 3 individual structures. So if we start at the bottom, we've got the medulla oblongata, the pons, and then we finally have the midbrain. And so those 3 things, medulla oblongata, pons, and midbrain, make up what we call the brain stem. So that's the structure. What's the function? Well, it really does 2 things. The first thing it's going to do are these more basic needs. It's going to keep your self-breathing, keep circulation going, digestion, swallowing. All of that is going to be controlled by the brain stem. If there's any damage to the brain stem, it's going to be catastrophic. What else does it do? Then we have information coming in. So we have sensory information, just like that worm did, coming up to the brain. And then we have motor nerves going out. And so the brain stem is important in routing that information and filtering that information, sending it where it needs to go. What's behind that? We have the cerebellum. The cerebellum, and the function of that is motor control. So as you do sports, for example, it's the cerebellum that's giving you that coordination. And it also gives you motor memory. So as you learn to ride a bicycle and you remember how to ride a bicycle, that's going to be thanks to your cerebellum. If we keep moving up, we now have the thalamus. The thalamus, again, sits right on top of the brain stem. How to ride a bicycle, that's going to be thanks to your cerebellum. If we keep moving up, we now have the thalamus. The thalamus, again, sits right on top of the brain stem. And so the best analogy I could come up with is a router. It's basically sorting data and sending it where it needs to go. If we were to look below that, there's a little structure here that's incredibly important. It's called the hypothalamus. It's going to be really right above the roof of your mouth. What is that accountable for? It's homeostasis. It's maintaining body temperature. It's maintaining osmolarity. All of that stuff is contained right up in the hypothalamus. Also important in circadian rhythms. And then if we look right below that, you can see a little gland hanging out. And 1/2 of that pituitary gland, the posterior pituitary, is technically part of the brain. And it's important in basically sending off hormones. And so there are nerves that flow into that pituitary, and it's sending out things like antidiuretic hormone that keeps your water balance the same. Oxytocin would be another important hormone that comes out there. If we keep moving up, then we get to the level of the cerebrum. What's the function of the cerebrum? That's integration. So what we're doing is making sense of all that data that comes in. Now, what makes up that cerebrum are going to be all these neurons. There's tons of neurons that are connected together, billions of neurons and billions of synapses or connections between these neurons. And that's where we're making sense of information as it comes in. Now, if you were to look at this image right here, so of that brick wall, so take a moment to look at that. I'm going to show you some other images. Now focus on this. And then that, and then that. And what we find is as you look at those images, your brain is integrating. It's making sense of all that information. And it used to be a black box. We didn't know really what was going on. But now we can use technology like a functional MRI, a functional magnetic resonance imaging. And what we're looking at here is a brain in action. So this same study was done on females. And what they would show them is something neutral, like a brick wall, and then a kitten, and then something like dirt, and then something like puppies. And so what we're seeing is as those images are switching back and forth, we can start to see where blood is flowing around in the brain, and we can start to figure out what the different parts of the brain actually do. We're able to figure out their function. So when we're looking at the cerebrum, every picture that I've showed you is from the side, so the eye's up here. But if we were to rotate that 90 degrees, now we're looking at it head on, we'll find that there are two hemispheres. There's going to be a right and a left hemisphere. Now they're connected in the middle using something called the corpus callosum. So that's a connection of nerves in between the two hemispheres. And we do tend to show lateralization. There are going to be certain things that we put kind of on the left side of our brain, like mathematical reasoning and logic, and things that we put on the right side, like facial recognition. Now, this is plastic. In other words, we can move these functions back and forth. And you can even have a radical hemispherectomy where you're cutting one of these out and you still have a function in your brain. Now, if we were to go right below the corpus callosum, we get into this area called the basal ganglia. It's made up of a bunch of nuclei. What are nuclei, or what is a nucleus in the brain? It's basically a bunch of neurons that are right next to each other that have the same function. And so all of these nuclei together make up what's called the basal ganglia. And you can see this would be the corpus callosum connecting it together as well. So this is below the cerebral cortex. What's the function of that? Well, scientists have been able to figure out this complex interaction of inhibition and excitatory response between these neurons, and basically it controls a lot of our motor control. And if you have somebody who has Parkinson's disease, then we're having problems in this basal ganglia area. As we move farther up the brain, we eventually get to the cerebral cortex, and that's going to make up about 80% of the brain. So it's most of the brain itself. And it's broken apart into these four lobes. And so if we start in the front of the brain, we have what's called the frontal lobe. What's the function of that? It's mostly executive function. So it's kind of like the boss of your brain. It's emotional control of it. And if we have people who have damage to that frontal lobe, they have really huge emotional swings. As we move back towards the back of the brain, we get to the parietal lobe. What's the function of that? Basically a sensation. It's you dealing with and reacting to your environment. So we have a lot of neurons coming in here from sensory input. As we move to the back, we have the occipital lobe. The function of that is vision, primarily vision. And then we move onto the side, we have what are called the temporal lobe. Temporal lobe is going to be important in language. It's important in hearing. It's also important in memory. We have a lot of memories in there. And so each of these lobes have different functions that are associated with it. And hopefully those little icons help you remember those functions. Now, if we're going to go inside the parietal zone, we find a really important part here. It's called the somatosensory cortex. And that's where sensory information is coming into the brain. And then on the other side of the lobe, we have what's called the motor cortex. And so going way back to that worm, we have information coming in, sensory information, and then we have motor output coming out. And so that's going to be a point of integration where we get information in, decide what we want to do with it, and then send that message back out. Now, if we were to look at that somatosensory cortex and map it along the cerebral cortex, we would find that we dedicate huge amounts of that brain surface area to things like your fingers, your tongue, your lips. In other words, we have way more neurons and way more sensory information coming in from your fingers, as opposed to, for example, your back. We don't have as much of it dedicated to that on the backside. We could also use functional MRIs and then even an operation to figure out where a lot of these things are located, like speech and smell and hearing. But over the future, we're going to get really, really good at figuring out specifically what are all the different parts of the brain, what are the nuclei, what do they do, and even mapping it down to the level of the neuron. So how did you do? Do you remember those 17 different structures and their functions? Well, it's time to review. So let's go through it. What's this one at the bottom? Overall, we call that the brain stem. Hopefully you got that. What are the three parts of the brain stem, though? Do you
pons, medulla, and pituitary gland.
remember that? Could you pause the video and then say what they are? Well, starting from the bottom, remember we have the medulla oblongata. We then have the pons. Then we have the midbrain. So
the midbrain. The midbrain. Never mind.
That's going to be the structure and where it's found. Can you remember the two functions of the brainstem? Two big things were, number one is to maintain breathing, heart rate, digestion, swallowing, so these fundamental properties of life. But what's the second one? Remember, it's to sort information going up and down. What's behind that? What's that structure called? That is the cerebellum.
Cerebellum, yeah.
And so what's the cerebellum?
It's stores like muscle memory, things like that.
Remember, that's coordination, motor control, and also
motor control.
So motor memory. Do you remember what sits right up above the brainstem?
Thalamus and hypothalamus.
That is the thalamus. What's the thalamus do? Remember, it sorts information as it
sorts information.
Moves up to the upper parts of the cerebrum. What's below that? That is the
hypothalamus, which is responsible for Memory, maybe long-term memory.
B, hypothalamus. Underneath that, what's that do? Remember, that's homeostasis. It's maintaining it.
Hypothalamus. Wait, I said it was also for memory, like memory and homeostasis.
Internal body state. Do you remember what hangs off the bottom of that? That is the
Pituitary gland.
Posterior pituitary. Hopefully you're doing well so far. If we keep going, then what is this upper portion of the brain
Cerebrum.
We call that the cerebrum. Okay, let's keep going into the cerebrum then. So do you remember what's that connection between the two hemispheres of the
Corpus callosum.
The brain? We call that the corpus callosum.
Corpus callosum.
And do you remember what we call those little nuclei that are found below that cerebrum? Those are
basal ganglia.
Called the basal ganglia, and they're really important in motor control. And remember, the corpus callosum allows our hemispheres to connect. If we were to go up to the upper portion, what do we call this? Highly folded upper portion of the cerebrum?
Frontal, parietal, temporal, occipital.
That's called the cerebral cortex. Do you remember what the front lobe is called? That was pretty easy. That's called the frontal lobe. What about the yellow lobe right here? That's called the parietal lobe. Do you remember what they do? Frontal lobe, remember, is executive or boss-like
functions. Touch and sensory
functions, and then parietal is going to be sensation of the environment. What about at the end? Do you remember that? That's called the occipital. And
responsible for vision.
And what about at the bottom? That's the temporal.
I don't remember.
Occipital, remember, is a location where we have vision, and then temporal is going to be more language hearing.
Oh, language. Okay.
Memories are there.
Language and what?
Other parts, we'll remember, is a location where we have vision, and then temporal is going to be more language, hearing, memories are there. Now, there are two other parts in our lobe. So what do we call this area right here, and then this area right here? Those are called the somatosensory cortex. Remember, that takes in information, makes sense of it. And then we have the motor cortex, which is motor cortex. So those are those 17 structures. If you don't remember them, you may want to watch the video again, maybe make some flashcards. But that's the brain, and I hope that was helpful.
Thanks. Thanks, Paul. Paul's a good guy, isn't he? Midbrain, hindbrain, forebrain. The forebrain includes the thalamus and hypothalamus, the basal ganglia, and the cerebral cortex. Okay. Again. Any plus of a cell, and this depolarizes a cell. You are leucine, Lu-L, nonpolar hydrophobic. You are glutamate. I think glutamate. Glue. E. Basic. Acidic. Sorry. Acidic, polar hydrophilic. pKa of 4. Yeah. Tertiary. Applies by hydrophobic interactions, acid base, hydrogen bonds, disulfide bonds. Side chain interactions. Folding patterns due to side chain interactions. Maybe I should just go again on that one. Forebrain. Postural stability. Smooth movements. Maybe the hindbrain. Maybe the hindbrain. Oh, never mind. midbrain, the midbrain. Oh, no, it's part of the forebrain. Raising smooth movement and helps postural stability. Five-brain cap and 3-poly, 3-poly 2. Sodium potassium pump and by leaky channels. The inner mitochondrial membrane. The frontal lobe, frontal cortex, frontal lobe. Speech, motor cortex, somatosensory. Oh yeah, somatosensory motor cortex, speech taste, reading, temporal lobe, smell, hearing, auditory. Oh, smell, hearing. Okay. The midbrain. Basal ganglia. Connect. Connect similar cells together. No. Yeah, I think so. No, I think it was something else. Oh, connect similar cells together? Connect adjacent cells together? No. Structure. Connect. I think it is connected. Oh yeah, hold similar cells together. Hold similar cells together. This one was kind of close. E plus S, ES, into E plus P. ES is K2, which is rate limiting step, catalytic reaction. And then we have ES going back to K. ES going to K1. That's K minus 1, and then E plus S is K1 towards ES. Binding of the enzyme to the substrate. What did I say was catalytic rate? I don't know if I said that or not. Basal ganglia. Oh, that's it. Not just a thalamus. Thalamus. Oh, midbrain. Oh, this is motor control. Oh, hearing. Gotcha. Okay. Oh, yes, hold on. The secondary structure of proteins. Hydrogen bonds? Oh, yeah, it's like hydrogen bonds. Proteins. Gray matter. The lobes. Cerebrum. Frontal cortex. Oh, cerebral cortex. Oh, okay. Basal ganglia. Outer. Hydrophobic interactions. Hydrophobic interactions. And there's others. Isoprene, ionic, hydrogen, disulfide bridges. Due to the cysteine interactions. Cholesterol. Cerebral cortex made up of the four lobes: frontal, parietal, temporal, and occipital. Touch, pressure, campaign, spatial processing. I'm gonna go with the parietal lobe. Yes. And then the somatosensory is in the parietal. The motor cortex is in the frontal lobe. The other ones don't have it, so it's okay. RNA polymerase comes in and starts breaking apart DNA nucleotides, creates a transcription bubble, and then begins copying DNA into RNA. that code for proteins. Translation. Elongation. Translation. So they're saying this is the um, hold on, let me see. Oh, okay. Forebrain. Tertiary structure proteins. Ionic interactions, hydrophilic. Big interactions, activation ratio, lysophil fibrages, hydrophobic interactions. Folding is made of interactions between side chains. Yeah. Nice. This is the frontal lobe, frontal cortex, part of the cortex. Cerebral cortex, frontal lobe. Fluoride ion sent to the neuron, and this inhibits the cell, but polarizes the cell. Oh, hyperpolarizes the cell. Not just polarizes, hyperpolarizes. Connects neurons, cap junctions. Reads the start codon. Scans. Sorry. Scans. Passive transport. Or blanketers. I don't know, buddy. What are you talking about? Oh, sensory processing. Okay. That makes sense. Chlorides enter the neuron and this hyperpolarizes. So, promoters. In which splicing is done, splice out the introns and keep the axons and add the tail and the 5' cap. Oh, done in eukaryotic cells. Yes. One replication origin, because of many, multiple. the occipital lobe, sensory opposite, sensory processing interactions, between the nucleic acid hydrogen bond loop. We're going to make up the shape, the RNA structures, secondary RNA structures we have, bulges, junctions, loops, helices. And the structures we have is helices, pseudonodes, and stem loops. Shape, loops, helices, bulges, and junctions. Electrons. Adjacent cells together by anchoring to the cytoskeleton. Found in cells that undergo a lot of mechanical stress. Cardiac muscle, bladder tissue, I don't know, gastrointestinal mucosa, epithelial tissue. I guess that's okay. Sensory processing occurs. Basal ganglia. Excuse me. Why must I say acetylcholine? What the heck? Not. GABA is the inhibitory, and what's the other one? Seriously? I keep thinking acetylcholine. Oh, glutamate. Not glutamate. And create a cascade of secondary messengers, right? Or then allow different signaling pathways, signal translation, protein kinase A, GPCRs. Okay. Yeah, GPCRs. GDP to GTP. Nature is probably so. This way. Cerebral cortexes, we have frontal lobe, parietal lobe, temporal lobe, occipital lobe. Glutamate. Yeah. Ribosome. Embedded. Ribosomes. The forebrain. The forebrain includes the cerebrum, the thalamus, hypothalamus, basal ganglia, what is it? Dorsal pituitary gland? Dorsal pituitary gland? I don't know. Let's see. Thalamus, hypothalamus. Oh, I don't think I said that. I don't really know if it contains a pituitary gland. Thalamus, hypothalamus, basal ganglia, limbic system. Limbic system? What's limbic system again? It does contain the pituitary gland. Oh my goodness. Keep dying to myself, huh? Pons, medulla, and what is right here? Pons, medulla. Pretty much it, huh? I think this is midbrain. Yeah, midbrain, just midbrain. Just a whole different part, just different structure there and stuff. Midbrain, thalamus, hypothalamus, pituitary gland, part of the forebrain. What? It's right there. Am I supposed to look at the limbic system? What is the limbic system? Instinct and mood, fear, pleasure, anger, hunger, sex, dominance, care, offspring. Okay. Alright, buddy. So can I not just have that? Like, I don't want it to be there. Why is it still there? We have to be in this one or something. Did I get it right? No, I don't think I did, huh? Okay, thalamus, hypothalamus, basal ganglia, pituitary gland, limbic system, I guess, and cerebral cortex. This is the parietal lobe, touch, pressure, temp, pain, spatial processing. And it contains the somatosensory cortex. The frontal lobe contains the motor cortex. Exocytosis? Okay. Voltage gated channels. Okay. N terminus and C terminus. Thalamus, hypothalamus, pituitary gland, limbic system, and cerebral cortex. Oh, I missed basal ganglia. Relay station for information, for the processing of information. Sensory information. I think that's good, right? Let's do it again. Ooh. I'm not sure. Oh, flip base. Flop base. Okay. Phosphodiester bonds. Primary. Motor. I said motor function. IQ. Motor function, right? Skeleton. Motor function. Smooth moves. Smooth movements. Postural stability. Smooth movements. Postural stability. Scans for circadian. Sensory information. Part of the forebrain. Thalamus and the hypothalamus. Part of the forebrain. It's like a little bit above the ear, huh? Okay. Chloride ion channels come in, which hyperpolarizes the cell. Occipital lobe. Do not go for proteins. Of large particles or large solid particles, materials. 30S leads on N4 methionine. N4 mil methionine. I think that's fine. Smooth movements and helps. Postural stability. Part of the forebrain. Executive functions, like regulation of emotions, things like that. Impulse control, speech and motor. Impulse control, speech and motor. Impulse control, speech and motor. Speech and motor. Separate units. Depends on mass and shape, and you can add these subunits. Or you can add the units. RNA polymerase. Tryptophan Y contains the indole group, emits fluorescent lines, aromatic, nonpolar, hydrophobic. Tryptophan TRP Y nonpolar hydrophobic contains the indole group, emits fluorescent lines. Projection areas are areas where sensory processing occurs. Okay. Cerebrum, where we have the frontal, parietal, occipital, and temporal. Allosteric inhibitor, such as non-competitive inhibitor. Non-competitive inhibitor, Vmax goes down. Km no change, and the x-axis touch on the Limburger plot. Frontal executive functions, speech and motor contains the motor cortex. Motor cortex. Yes. Impulse control. Through an unwinding of chromatin. video from him. Unraveling and winding. That's fine. Yeah. Telomeres are shortened during replication, but it can be reversed. Reversed. Oh, I see. Reverse. That's why I mean reverse. Released by one particle moving down its electrochemical gradient, driving another particle up its gradient. That's fine. Lots of different senses, mostly to deal with like touch, things like that. Touch. 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 Touch, pressure, temp, pain. Special processing. I'll do it again. Dissolved in water and soluble in non-organic. Organic non-polar solid. Glutamate. Phospholipids of a methylated guanine, a bunch of adenines. Yeah. Which part signals ribosome attachment? Oh, the 5' cap does? Touch, pressure, temp, pain, and spatial not spatial movement, spatial processing. Yeah, what processing is. GABA. Cleavages. Usually will form double bonds or ring structures. Decarboxylase. DNA wrapped around a histone protein core, H2A, H2E, H3H4, which are connected by an H1, which is DNA. It goes through an H1, and then that's how it wraps around the histone core. Yeah. Succession of tRNA adds their amino acids as the mRNA is moved through the ribosome one codon at a time. Touch, pressure, temp, pain, and movement processing. Spatial processing. I think it missed pressure. Forebrain includes the thalamus, hypothalamus, limbic system, basal ganglia. Oh, and the cerebral cortex. Oh, jeez. Cerebral cortex is projection areas. Flip-base, flop-base. Prokaryotic mRNA that encodes multiple proteins. Eukaryotes, it occurs in the mitochondrial matrix. Prokaryotes, it occurs in the cytoplasm. Glycerophospholipids. The occipital lobe is associated with pressure. Well, like I guess, touch. There's a touch, pressure, temp, pain, touch, pressure, temp, pain, and spatial processing. Oh no, it's the occipital lobe. Oh no. Bridal lobe. Okay, bridal lobe. Touch, pressure, temp, pain, spatial processing. Okay, yes. Finally. Thalamus, hypothalamus, pituitary gland, limbic system, and the cerebral cortex. There we go. These are called projection areas. Sensory processing. Sensory information, part of the forebrain. Results in one gene coded for multiple proteins. Helixes, secondary. Oh, beta plate sheath. Prokaryotic translation. Prokaryotic translation. Inhibits negative sense. Aniguanine 1. Oh, no. Oh, man. Occipital lobe is associated with vision, just vision. 8 for sensory information. It's part of the forebrain. Controls motor functions and helps smooth movements and helps spatial processing. No, smooth movements and postural. Federated units, Prokaryotics have 70 S, which is 50 and 30, and eukaryotics have 60 and 40 S, 80 and 60 and 40 S. Cerebrum. Smooth movements and spatial processing, spatial movements. Postural. Oh my goodness. Executive functions. Motor, related motor cortex, speech. Okay. Impulse control. I miss impulse control. I think that's important. Smooth movements. smooth movements and oh man, seriously. Again, seriously. Postural stability. Why can't I remember that? Okay, executive functions, emotional stability, right? Or something. Yeah, I think emotional stability. Emotional stability, motor, contains the motor cortex. Impulse control, and I forgot speech. Smooth movements, postural stability. Yes. Executive function, speech, motor, contains the motor cortex, and impulse control. Yes. GABA, touch, pressure, pain, touch, pressure, pain, and spatial processing. Yes. Thalamus, hypothalamus, pituitary gland, limbic system, and cerebral cortex. Oh, I forgot basal ganglia. Projection areas. Thalamus, hypothalamus, basal ganglia, limbic system, cerebral cortex, pituitary gland. Okay, purines have 2 rings, adenine, guanine. Okay. Beta sheets, ripple strands, reveal or integral are common in secondary protein structures. Yes. mTOR inhibits vision, sensory information found in the forebrain. Smooth movements and postural stability. Executive functions, speech, motor, impulse control, contains the motor cortex. Thalamus, hypothalamus, limbic system, basal ganglia, pituitary gland, and cerebral cortex. Oh, let's go! That took way too long, though. That took way too long. Hey, how's it going? Unavoidable garbage. Hey, Julianne. Hey, guys. How's it going? Everyone's doing well. Is that a Chihuahua? Is that a Chihuahua? Oh, no, it's not. Okay. What a name. Unavoidable garbage. All right, well, I'm going to take a 10-minute walk around. I think you guys need to get some more blood going. Let's not start it yet because I don't want to mess up my time. Anyways, I'm gonna just walk around in the backyard, get some blood going, and then let's finish up Anki. I want to review some gaps, take a little bit of time, and then we'll go ahead and start. And then we'll go ahead and start The next one after the review, you know? Yeah. I wonder if there's anything here and how you. Anyways, yeah, I'll be back. You can see this one now. This one. Today's workout was wonderful. Okay. Sorry, guys. I had to go to the bathroom. Okay. Let's get started. Yeah. Substrate concentration. Neuronal crest, right? Neuronal plate. I was gonna edit this one. Yeah. I don't need this one. It's already in there. So let's actually edit it. Remember the lungs. I think that's better for me because urinary bladder I already know. So let's do that one again, I guess. Second stage is S phase, synthesis phase. Sure, enzyme or ES complex. Oh. Ah. Ah. The average of the two pKa values. Lungs. No. Lungs? Yeah, lungs. Ectoderm. Ectoderm is the attractoderm. Associating everything you find attractive. Associating everything that you find attractive. Yes. Yes. Good. Diploid cell. Meiosis. The neuronal plate, maybe? Oh, no. Neuronal. Oh, maybe the neuronal plate? Neuronal tube? Oh, neuronal crest. Oh, my goodness. Now when it's neuronal crest. I can't believe you've done this. I can't believe you've done this. Zygote. Zygote. Crossing over an independent assortment. Germ cells. Well, if it's prepared for cell division, then it's a G2. Let's go. You guys see this? Yeah. Garbage. Interesting. Two strands of DNA, both strands. Neuronal crest cells. The neuronal crest cells. Meiosis. So when you really think about it, that means that pollen. That means that pollen is hello. So hello. I think it's so close to it. Hello. That's fine. Anyways, I guess when you think about it, that kind of means that pollen is. Yeah. Well, that's fine. I'm not gonna say it. I don't think I'm gonna say it. I'm gonna say it. I don't care that you broke your elbow. Really good pancreas. Identical. Diploid. Mitosis. Not this again. Dun 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 dun. Stomach, urethra. Yeah. Zygote. Zygote. Endoderm. Not. Mesoderm. Not. Endocrine. Wait, no, endocrine. Yeah, autocrine. Sexual reproduction. Crucial for offspring. Genetic diversity. Okay. Starting with 23 1N. Let's put on some lotion after this. I don't know if you guys can hear me anymore. Really don't know how the microphone works. Whatever. Against firm cells. Yeah, this usually against firm cells. No. Replicating DNA, replicating chromosome. Mm-hmm. Specialized regions within the mesoderm include the what are you talking about? Oh, the nodal cord. Gametes. Germ cells. DNA ligase seals the phosphodiesterase backbones of newly synthesized DNA. Yeah, repaired, I guess. Ectoderm. Endoderm, not mesoderm. Max. Look at this. Gallbladder. Gametes. Nodal cord. The nodal cord. The development and thickening portion of the ectoderm forms the neuronal tube. Wait. No, please. Okay. Mesoderm, endoderm, ectoderm. Churn out the ribosome immediately into RER. Lip guts, liver. skin, hair, nails, face. Oh, skin, hair, face, skin, eyes, maybe. And then the nervous system, brain, spinal cord. I think maybe nervous system is brain, spinal cord. So yeah, right. Teeth, the lungs. I feel like it's like the same cars over and over. Like 20 cars are the same thing. After a growth phase, so that's G2, right? Termination factors. Two identical daughters dividing, division. Diploid cells, diploid division. What are we talking about? Cell division. 4? So, 2, 2, 1. Oh, 1, 2. What the heck? Ectoderm. Ectoderm. Notochord. Oh, oh, oh. Oh, ectoderm. I thought that's from the notochord. Never mind. Egg cell. Reduction. Sperm cell frozen egg cell. Where we have organelle and membrane replication. Well, I guess replication. Maybe more production, right? I don't know if they enter replication. Yeah, that's actually very interesting. I don't know about that. Well, producing membrane organelles. Neuronal cross cells. Where did I go? Kidneys. Yeah. Ectoderm. Liver, lungs, pancreas, gallbladder, urinary bladder. 2 sets, 46 chromosomes. Well, we're getting there. It's going down, I guess. I guess it's just super slow. Lip guts. Production of organelles and cell membrane. Production of organelles and other cell membranes. Organelles and what? Membranes. Membranes? I guess, yeah. Oh, membranes. Okay, yeah, cell membranes. Yeah, that's fine. Zygote. Urinary. Pancreas. identically structured, but have identically structured and code for the same gene, but have different alleles. Body cells, mitosis, undergo mitosis, diploid. Blastocysts. Blastocysts. And upregulate and regulate function. Um, I was gonna say babies and sugar, but tyrosine tyrosine. I want to say methionine 309. Problematic effect. Oh, 309. Let me see methionine real quick. How does methionine look like again? Ah, not methionine. What the heck? What am I saying? Definitely not. Definitely not methionine. Serine though. Serine, yeah. Yeah, okay. Yeah. Yeah, okay. 23 in, 23 there, 23 1 in. Endoderm, not mesomere. G1. Six comes off top. Genetic material from both parents. tRNA. Aminoacetyl tRNA synthesis. Gastrulation occurs. Mm. 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 Second week. The nodal core. Oh, that's in the mesoderm. I didn't know that. Mitotic, somatic cells. Mitosis. neuronal plate. Q is higher than KEC. Here we go, price to reactions. (23N), 23(1N). Oh, facial bones, the adrenal medulla, Swan shows melanocytes. You right there, Belada? The glands. Stomach. Liver. Intestinal epithelium. 23, 46, 2N. Oh, well, 2 sets of chromosomes. Lagging Okazaki fragments. Derm, mesoderm. Ectoderm, mesoderm, endoderm. Schwann cells, melanocytes, facial bone structures. I forgot the other one. Adrenal medulla. That's fine. Teeth, face, eyes, hair, nails, brain spinal cord. Somatic cells. Meiosis, but not in mitosis. Longitudinal axis. Zygote, sexual reproduction, fertilization. Juxtacrine. Longitudinal axis. (46). Where we prepare for mitosis meiosis. Yeah, well, we prepare for production of organelles and cell membrane. Right? Ectoderm. Excuse me. Blastocyst. Upregulate or downregulate the function. Interesting chemicals related include serine, 309, and tyrosine. Also, pneumatic effect. Fertilization is the only event that changes the chromosome count, ending with 46. Around the second week, 2 sets. Schwann cells, melanocytes. I don't know. Facial bones. Facial bones. Something medulla. Schwann cells, melanocytes. Adrenal. Facial bones, adrenal medulla, Schwann cells, melanocytes. Yes. Facial bone, adrenal medulla, Schwann cells, melanocytes. We got someone this one. I don't know. I think maybe the hindbrain. Hindbrain for sure, right? That's forebrain. What the heck? What the heck? Never mind. Hypothalamus. Hippocampus. Oh, I got it mixed up. Hippocampus. Thalamus and homeostasis. Thalamus is Schwartz theta. Aha. Homeostasis. Okay, sure. Gotcha. Higher motor neurons. Things like consciousness. What is it? Consciousness, awareness, thoughts or something. Thinking, thoughts, awareness, consciousness. Something. Cognition, emotions, cognitive. Whomst. Does that stand for something? Whomst? What does that mean? I don't know. It does or not. Cognition, emotions, consciousness. I think it's fine. Hypothalamus. Yes. Okay. Hyporeflexia, overextension of the muscle reflex. Decrease muscle stretch. It decrease. Oh, hypo. Hypo. Lower motor neuron sign. I wish I could zoom in a little bit more. Mm-hmm. A decreased muscle stretch reflex. Or action potentials to excite, to contract, to stretch. Okay. Hammer hits the patella tendon, stretch receptor, or neurons, sensory neuron. Fire cell neuron dorsal root ganglion. Sensory neurons always enter towards the backside. And that is the gray matter. The inside, white matter on the outside. Opposite of what the brain is. So the brain is gray matter and white matter on the inside. But anyway, so dorsal root, right? So sensory neuron, dorsal root ganglion, enters towards the backside of spinal cord, the dorsal side. Interneuron, it's the body of the motor neurons. That gets sent up to the brain. We have efferent neurons here coming back, and it's going to contract. Okay. Decrease in the muscular response. A decreased muscle stretch reflex. Just you have to know this. I guess. What is that? Oh, that was a good pop. Somatoneurons efferent in the muscle spindle synapse. In the spinal cord, as the stimulus axon gets sent back out to the lower motor neuron. from higher motor neurons from brain to contract the muscle on top of the leg. The quadriceps muscles on the bottom are inhibited. So it tells the quadricep muscles to fire, to contract, and then it sends out another signal. Well, when it sends out another signal, it's all going to be from the front, right? Come back down, and then it's going to send it out from the front, and it's going to say, okay, go ahead and inhibit these muscles in the hamstring. All right, I think that's okay. A decreased contraction in the motor reflex test. Muscle stretch reflex. Muscle stretch reflex. Send signals out to the lower motor neurons to activate, to control lower motor neurons. Motor nerve cells, upper motor neurons. Motor nerve cell, upper motor neurons. Down spinal cord. Tell the lower motor neurons. Yeah, well, activate organoids for most of the brain. Control lower motor neurons. Okay, so we're gonna have the hammer hit the patellar tendon. That's gonna send a signal through from the nerve cells, from the PNS, from the somatic. Oh wait, don't we have two types of cells? Somatic. Sensory cells, sensory receptors, up to afferently on the dorsal side, of the nerve that travels to that site of the particular vertebrospinal cord column that controls that site, and then it's going to most likely be sent up to the parietal lobe, part of the forebrain, part of the cerebral cortex, parietal lobe, associated with touch. Go through as it gets up to there, it's going to travel through to the thalamus. It's going to process information, go up to that cortex, come back down from the higher motor neurons, send it back down efferently through the efferent neurons, and then exit out towards the front, towards the ventral side, front of the spinal cord column side, and then it's going to send out the signal, electrical potential to contract the quadriceps, but also maybe send glutamate neurotransmitters, but also sending GABA inhibitors towards the what is the other part? Towards the hamstrings, and that's going to cause a knee-jerk reaction. Let's go. Oh, gray matter. Gray matter is inside. White matter is on the outside. Oh, and I forgot to talk about the interneurons. That's fine. A decrease in the muscle stretch reflex. Let's go. Control lower motor neurons. Let's go. The thalamus. Oh no, the hypothalamus. The hypothalamus. Upper motor neurons are found in the forebrain and synapse on. Not sure. The lower motor neurons in the brain synapse spinal cord. Okay. An increase in the muscle reflex. Muscle stretch reflex. I guess. Test. Oh, I think it works. Yeah, I think it's anyway. The forebrain becomes cerebral cortex, cerebrum, contains the hypothalamus, pituitary gland, limbic system, and cerebral cortex. Frontal lobe, parietal lobe, temporal lobe, occipital lobe. Okay. CNS, PNS, automatic, and just automatic, parasympathetic, synthetic. Oh, I'm not even reading the question, dude. Where's my head at? Where's my brain at? Are found in the brain and synapse on the spinal column, lumbar, spinal cord, brainstem, cerebral cortex. Homeostasis, so hunger, sweat, thirst, etc. Things to keep us in homeostasis, right? Contract after it's stretched too far to protect it. consciousness, emotional regulation, was impulse control or something, and things like that. Just things like that. Emotions. Cerebral cortex, synapse on the spinal cord. Yeah, all that. I mean, what I said before. I'm not gonna talk about it again. An increase in the muscle stretch reflection. Yeah. Oh, hypo. Control the function of the element. Decreased function in the muscle stretch reflex. Increased function in the muscle stretch reflex. Thirst, hunger, anything to do with homeostasis, etc. Autonomic endocrine system controlling biological trends. Cerebral. Emotion, cognition, consciousness. Cerebral cortex, synapse on the spinal cord. Decrease in the muscle stretch reflex. Three, 5, 2, and 2, which is 7 and 9. I think that could work. That would probably work. Excuse me. One and one. Excuse me. Humoral, maybe? So medium. Dang. Dang, they got me. They got me. I think it's gonna take too long for the review. There's only a couple of things that I think I need. I think I might have went over a little bit too fast and I want to go over. Brain stuff, I'll be okay, I think. Oh, I still have to do this one. Actually, no, I kind of like it being out. Go back, go back. Go back. Yeah. Stay there, I think. Also want to go through the tabs, figure out what I want to delete. Later on, though. Not right now. Go again. I shaved. I keep having to shave. You know? Like, cause I joined the Air Force, and you're not allowed to have facial hair, but the facial hair grows pretty fast. So I don't know. I'm already picturing now I'm gonna get yelled at by the training instructors they call them. Did you shave? Einstein, did you shave? Yes, sir, I shaved. Why do I see so much hair? It goes back so fast. Go back and shave. Sir, yes, sir. Inner and outer membranes. Ribosomal synthesis. Wait, what? Okay, nucleus inside of DNA replication, DNA transcription, post-transcriptional modification, three phospholipid bilayers, inner and outer, contains the nucleolus, which is where ribosomes, therefore ribosome synthesis occurs. Oh, I think RNA, our RNA is created as well, right? RNA. Oh, let's go. Okay, so it's a cell-to-cell immunity, right? Cell-to-cell immunity. I think it's the same thing, right? I think it might be the same thing. Cell-to-cell immunity. Cell-to-cell. Maybe not. I think so. We'll go again. All right, there we go. Carboxylic acid backbone is 3. Amino backbone is 8. Aspartic acid is D. Glutamic acid is R. Erectile dysfunction. That's how I remember it. Erectile dysfunction. Erectile dysfunction is ED. So both are 4. Histidine is 6. And LA, lysine, and arginine. Lysine 11, arginine 12. That's fine. Parasympathetic. Whoa, buddy, that's a lot of text. But I'll read it. I have to read it. Okay. Physiological response to the sympathetic or parasympathetic nervous systems, rather than just the terms themselves. Excuse me. They're sympathetic. Halt on gastrointestinal peristalsis, which I think it just means breakdown of food or like digestion, you know? Just another fancy word for that. Increase blood to the skeletal muscles. Increase heart rate. All right. Inhibition of digestive enzymes. And then opposite of Sympathetic is the stuff that's going to be parasympathetic. Be able to link an intruder coming to your home with pupil dilation and salivary inhibition. Okay. You got it. It mostly just comes from the thoracic vertebras. Vertebrae. I think vertebrae. Vertebrae. Oh, vertebras if it's more than two, right? Or more than one? Whatever. Anyways, interesting. It's coming from thoracic, T1 to T12, sacral. What are the top ones? Neck. I'm thinking neck, but it's not neck. It's something or whatever. And these are coming from the stem, the brain stem nerves. Some mediated immunity. Oh yeah. All right. Let's phenylalanine, Phe, PHE, phenylalanine, Phe, nonpolar, hydrophobic, phenylalanine, Phe, wife, F, wife F, nonpolar, absorbs UV light. I don't think it's on there. That's fine. Let's take a break. Hey, Chucky. How you doing, bro? Yo. What's up, bro? What's up? How are you? We're on the last. No. No, I'm okay. It's not that bad today. What are we at? What's the weather at today? We're at 78. We're at a nice 78. Look at that. We're at a nice 78. Cool 78. No, I'm fine. I take a shower every night. I'm a night person type. A shower. I take shower at night. It's been a while since I took showers in the morning. That was when I was like a kid at school. Now I'm an older man. Older? No, not even. Not even. No, I could study more. Ideally, really, I should be hitting 12 hours each time on every single video. But, kind of started to think about gaining some weight and stuff like that, and I think that's affected like my energy and things like that. But I don't know, it's probably just an excuse, right? I should start running. A lot. Fidget toy. Oh, that's fine. I forgot about the fidget toy. I'm not even doing anything. Nah, sometimes just play with the pen. That's alright. Don't worry about it. No, that's alright. Don't worry about it, Chucky. You're good. You're good, bro. Yeah. No, you're fine, brother. It's okay. Don't worry. You're good. No, it's okay. Yeah. Today's gonna be a good day though. Today, I don't know. I feel great. A chewing habit? Maybe. I don't know. Actually, I need to talk to my dentist. I lost the dentures. The dentures? No, that's not the biggest dentures. What is it called? Dentures? Is it dentures? Retainers. I lost my retainers. No, I never bit my nails. I'm not that type of person. Pens, sometimes. I think I'm only doing it now mostly because I think I'm just stressed. I think I'm just stressed. There's just a lot going on, and I need to do well on the test, and I have other stuff too, um, like I'm taking 2 classes as well, pretty soon. Some college classes. I'm taking anatomy, human anatomy, and physiology. I took one, but it was a combined course, and I'm just not sure if the schools are going to count that. So I just kind of had to hurry up and find some, which I did. So I'll start those in June. So I start that very soon. They're summer courses, so they're pretty fast paced, and kind of worried about how that's going to affect the studying for the MCAT. But you gotta do what you gotta do. Do you have pens have bite marks? That's the way to tell if you're stressed. I mean, no, I could, but no, they don't. I mean, they would, but I think I'm just conscious that I don't want to actually ruin them, I don't want to have to buy new ones. And I also don't want to eat the plastic. So part of me wants to, but I'm kind of stopping myself. If that makes sense. Hey, yeah, this is pretty cool. You guys are gonna watch me take well, no, I mean, I'm studying now. No, I'm good. I'm alright, Chucky. Thanks. They're good pens. These are very nice pens. I don't know if you can see. Look at the pens. They're Japanese pens. Hold on, let me get the background. It's not focusing, huh? Focusing on my fingers. Anyways, yeah, then just use cheap ones. No, no, I can't. I mean, I can. Well, I can. Oh, these aren't too bad. Well, maybe they're kind of expensive. They're like $10 for the set. They're nice pens though. I don't want to ruin them. I don't like. No, these are the best pens I found. Ever, to be honest, ever. I really like these pens. These are. I mean, the cheaper ones are the Bics. The Bics. Still not focusing. The Bics. Can you even see? The Bic. Actually, how do people do this? This is kind of hard. Whatever. Anyways, no, I'm alright. Thanks, though. Don't worry, Chuck. Don't worry, Chuck. Thanks. Thanks, buddy. That's pretty cool. You guys are gonna. Yeah, I was just thinking about. You guys are gonna watch me do quite a bit of stuff this year, to be honest. I mean, MCAT, submitting applications, going through 2 classes, physiology, human physiology, human anatomy, take the MCAT, and then I'll shoot a video too when I come back from basic military training, if all goes well. Hopefully I come back in November or something. Anyways, I'm gonna go for a walk. I gotta get some blood. I sit down a bit too much, and I don't think I'm gonna have any time to. I don't think I'm gonna have any time to exercise today. So just a quick walk. I'll be back. Okay. My bad, guys. I got caught up talking to the neighbor. Hold on. Hold on. Let me go restroom real quick and then get some ice cream and then we'll come back. Okay. Oh, my legs are dead from from the weight and workout we did maybe 2 days ago, I think. 2 years ago now. Shows you have to come here. size, structure, shape. Oh. Size, chart, structure. Some medium. Paris, medium. Add. Search rate. Lower the memory potential. And plus? Okay, plus. Oh, actually, here the NH valves will shut off. And the iron channels. Voltage gated channels. Cytoplasm. Anticodons. Half the Vmax. Active potential. He might have a video on it. I should watch some of his videos. John might have a video on it. But he doesn't want to watch it. A plus. Potassium. He said it's 55 though. Tryptophan. W. Nonpolar. Emits fluorescent light. contains the indole group. Yeah, that's fine. Right, those. Sodium potassium pump. Mm-hmm. Mm-hmm. I shouldn't have had the ice cream. Whatever. Two pair. Yeah, 2 pair. Extracellular. Intracellular. Mm. That's why I got confused with the other one. I don't think I'm near you with this, to be honest. I don't remember us talking about tropomycin. I don't think so. Unless I missed it, but I don't know. I just really don't think so. Troponin. I think let's suspend, to be honest. I don't think I'm here. Haploid. Automatic, somatic, and then somatic has motor and has sensory. And I think so. Automatic, autonomic. What? Oh, okay, sure. Yeah, motor. So CNS, PNS, somatic, autonomic, parasympathetic, sympathetic, PNS, somatic, motor neurons, and sensory neurons. Yeah. Somatic's voluntary stuff, and then autonomic is involuntary. Yeah. Parasympathetic uses acetylcholine, and sympathetic uses epinephrine or norepinephrine. Stop coding this. Non-spontaneous or delta G prime. Yeah, it's gonna have a positive value. And oops, somebody messed it up here. There you go. Innate? No. I think it's innate, right? Not specific, fast, no memory. Innate. Oh, let's go. Skin, stomach acids. It's not main process. Keeps taking us back to this video. We'll watch it, but we're not in the immune system yet. Size, charge, structure. Ratios. No, concentration. Well, concentration. Isoleucine, illa, I, non-polar, hydrophobic. Sympathetic. Other people's. Enthalpy, positive delta H, heat consuming, and endothermic. Yeah, delta G is equal to delta H minus T times delta S. So minus the energy bond enthalpy minus E. H, delta H is equal to E plus P times E. To stimulus? Well, fire actually. It seems force known to threshold potential. Forces known to threshold potential. Oh, right. Okay. Yeah. What am I thinking, dude? Ion slip into the neuron, raise the voltage inside the neuron, away from the rest of the tissue, makes it more positive. Again. Independence. And multiply. Actual potential. Wow. Tropomycin. Calcium. Suspend this one too. K plus. Threshold potential. Nitrogenous base. T cells. Multiple codons. Unambiguousness. Degeneracy. One of the two. Oh, we gotta see. Glycoprotein. Phospholipids. Phospholipids, protein, lipids, carbohydrates. Yeah, phospholipids. Mitochondria. Sodium, potassium. One amino acid. Repolarization. Competitive, uncompetitive, non-competitive. Yeah, well, sure. What is this? I think we were watching this one. Guanine is complementary to cytosine. Cell mediated. So I said intracellular, like viruses and cancers. Oh, we're still not here yet, to be honest. Are we going to be here? I don't know. I think we will. Let's just spend it again. AUG, methionine. The potassium channel is open while the sodium channel is closed. Yeah, there we go. Equilibrium. Rate, kinetics. And autonomic. Protons. No specificity, non-specific, competitive, uncompetitive, non-competitive. Extracellular pathogens of bacteria. Receptors. Troponin. No, they were very either. loosely condensed. Less finely condensed. When the memory potential is reached, shouldn't it be the same thing? Sodium channels. I think maybe potassium channels. Potassium channels, yeah. Into point. The process is slow. Somatic. Smooth ER, lipids, hormones, that's all. Mitochondria, non-competitive, E or ES, effect on KM, no change, effect on Vmax, decrease, bias on surgery, yes, can added saturate or inhibition, no. Innate immune system. Non-specific, associated speed, very fast, right? Very fast. Concentration, WAG, UAG, not, no memory, WAG, WAG, sympathetic, epinephrine, norepinephrine to the threshold potential. Repolarization, and then it takes time for it to close. And that's what happens here. But that's closed somewhere here. I'm guessing right below the resting potential. It's just slower to close, and then sodium potassium pump brings it back to the resting potential. Okay. Cell mediated uses T cells. Decrease in mitochondria. So when peak is reached, K plus channels open, they rush out, repolarization, while the sodium channels will close. VNS is divided into that nurse. Okay, somatic, autonomic, no specificity, resting membrane is reached. Okay, yeah, that's what I just said. So when resting, the K plus channels begin to close very fast. Mitochondria, no memory. All right, let's take just a quick, maybe 5 or 6 minutes before we get started on. We'll finish up the synapses, and then we'll do some review stuff that I want to do for maybe like 30 minutes or 40 minutes or something like that. Once we wrap that up, then we'll start the next one. We'll do endocrine and respiratory today. Oh, sorry, endocrine and circulatory. There we go. We might watch, after we finish them, we might watch some of the videos if Andrew K has any more. I'm pretty sure he does, but we'll see if he does. Anyways, let's go ahead and take that break now. Do you do cars every day? No, I haven't hit cars yet, Nadine. My friend told me once I hit cars, well, I mean, I've been kind of doing cars from the passages, but like once, I don't think there really is a car. Is there a car reviews? I don't know. I don't think so. I don't know. I don't think so. Is there? Let me see. There's some practice passages. I might do some practice passages here, to be honest. I don't know. I kind of really like the setup from Khan Academy. Like, it's really nice. No, I haven't, but I'll hit Unit 5. And just do some of these practices. What I like about it, excuse me. What I like about it is that it's very similar to well, I don't know. I mean, I haven't hit this yet, but from the passages, I've been doing okay, to be honest. So, no, but we'll do Unit 5. Passages. Read and then just do yeah. Huh? Do it after. So does not accurately describe synapses. This is a true. What the heck? Electrical synapses? I think that's true. I think C. Maybe A then.
And we use synapses to do that. We're mostly going to be talking about chemical synapses in this video. In a chemical synapse, you have an action potential move down an axon. It triggers the release of neurotransmitters into this gap or this cleft right here. They're going to dock with chemical gated channels on the other side, and that can lead to an action potential on the other side. Some early work done with neurotransmitters within an axon. It triggers the release of neurotransmitters into this gap or this cleft right here. They're going to dock with chemical gated channels on the other synapses to do that. We're mostly going to be talking about chemical synapses in this video. In a chemical synapse, you have an action potential move down an axon. It triggers the release of neurotransmitters into this gap or this cleft right here. They're going to dock with chemical gated channels on the other side, and that can lead to an action potential on the other side. Some early work done with neurotransmitters was done by Otto Lowy. He had this dream of a wonderful experiment in the middle of the night dealing with the heart of a frog. He wrote it down, but when he woke up in the morning, he couldn't read his handwriting. Thankfully, he had that same dream again, and this is how the experiment that led to a Nobel Prize winner. He had the heart of the frog that was beating, and then he would stimulate the vagus nerve in the middle of the night dealing with the heart of the frog. He wrote it down, but when he woke up in the morning, he couldn't read his handwriting. Channels on the other side, and that can lead to an action potential on the other side. Some of the early work done with neurotransmitters was done by Otto Lowy. He had this dream of a wonderful experiment in the middle of the night dealing with the heart of a frog. He wrote it down, but when he woke up in the morning, he couldn't read his handwriting. Thankfully, he had that same dream again, and this is how the experiment that led to a Nobel Prize win. He had the heart of a frog that was beating, and then he would stimulate the vagus nerve on the surface of the heart, and what that does is slow the beating of the heart. So we're sending a message to the heart. He then took another heart from a different frog, but he removed the vagus nerve, so there's no neuron there. He got it beating, but he moved some of the liquid from the stimulated heart to the non-stimulated heart, and it slowed down as well. What did that mean? There was something in the liquid. He discovered these neurotransmitters, and he discovered a chemical synapse. In a chemical synapse, the action potential moves down a nerve, down a neuron. It triggers the release of neurotransmitters into the gap that are going to dock with receptors on the other side. Now, that's only one type of synapse. We also have electrical synapses, and in an electrical synapse, an action potential is going to move down. And again, that's triggered by these voltage-gated channels. Once it gets to that synapse, it's going to open up more voltage-gated channels, and the message is going to go. An electrical synapse, if you have an action potential in the neuron before the synapse, you're going to have an action potential in the one after. What's great about electrical synapses is that they're very, very fast. What's bad about them is that there's no control. So let's talk about chemical synapses.
No control.
To identify the different parts of it. So this would be where the information is coming in. So on this neuron, and this would be the cell body of the adjacent neuron. So the presynaptic side and a postsynaptic neuron. And then this little gap in the middle is, so on this neuron, and this would be the cell body of different parts of it. So this would be where the information is coming in. So on this neuron, and this would be the cell body of the adjacent neuron. So the presynaptic side and a postsynaptic neuron. And the little gap in the middle is going to be synaptic cleft. Now, there are structures that hold this in place. It's not just floating there. On the inside of that terminal bud, we have synaptic vesicles, and those are filled with neurotransmitters. So there are different chemical messages that can be sent across that synaptic cleft. There's some other anatomy that we should cover. There are going to be voltage-gated channels on the terminal bud. Those are going to be calcipoling, so a video that I made on those vesicles. They're going to activate protein. So they're sending a message very important in the transmission of that action potential for a neurotransmitter. A neurotransmitter releases potassium or powder chloride in, and they're stopping. Remember things early from this video. A lot of that has to do with connections in neural pathways in your brain. And what we're starting to find is that when you really remember something, that as we fire action potentials, as we have more of these synapses being activated, what we have is long-term potentiation. So if you look at what's going on here, if we keep firing this neuron, we've done this in the lab, we keep firing and releasing neurotransmitters on the postsynaptic side, the cell will actually develop and build more of these receptors, so it's more susceptible to that. And so we're building pathways of memory over time, and that's really what memory is. It's a set of neural pathways that are firing inside your brain. So that's the chemical synapse, and if you can remember a lot of the parts on this pre- and postsynaptic side, you're well on your way to improving your memory, and I hope that's
Pretty informative video though. Man, I'm happy we got the Anki done. We got to keep going though. Anki every day. Anki every day. Anki every day. Anki every day. What was I doing? Oh yeah. So, okay, electrical synapses. Okay. That's true. That's true too. Okay, cool. What channel plays a role in the release of neurotransmitter into the synaptic cleft? The voltage-gated calcium channels. Which of the following statements about action potentials and information transmission in the synaptic cleft is false? So we're looking for the false. That's fine. So, which of the following statements about the action potentials and information transmission in the synaptic cleft is false? Okay, let's see. So we're looking for the false one. This is true. Oh, wait. Frequency of an action affects the amount of neurotransmitter released. Naturally, the other one. Actually, more calcium. Actually, there's one more calcium leaving the target cell. Leaving the target cell. Leaving the target cell. Leaving the target cell. I think this makes sense. Maybe A. I don't know. Okay, what is the most common inhibitory? GABA. Good old GABA. Gamma, mama. Boom. What neurotransmitter is not classified as a catecholine? Catecholine. Seriously? Let me see if I can remember the structures. I think, there's another. I mean, we had, how many did we have? I think we had 5. Of those 5, only these 3. And we missed out 2. And I think, I don't think he was dopamine. These all kind of seem to be related, right? So maybe histamine. Yes. There are 2 types of neurotransmitter receptors. Which of the following statements about receptors accurately describes how they function? Okay. Ionotropic. Is that true? And this seems true. I think this is probably. I think this is true. This is true, I think. Well, actually, maybe. Ions don't pass through the membrane. That's false. I don't think so. I don't know. I think the one I got is C. I could be wrong though. Ah. Oh, okay. I thought they did. They don't. Oh, maybe not. Actually, I don't think they do. I think it's just a chemical synapse. Never mind. What am I thinking about? What am I thinking about? I'm so maybe D then, or what happened? Maybe D. So okay. Let's see. Oh, metabotropics are the ones. Okay, so I got it mixed up. Wow. I can believe you. Completely got it mixed up. Okay. I don't like writing it down on there. I stopped doing that because it gets erased when I move on to the next question. So that's just kind of a waste of time. Okay, let me read it. Okay, no, let me read it. Oh, okay. Why that right there, right? Most important. Oh, really? Synapse location? The synapse location? Well, I really think it would have been the type of neurotransmitter. Presynaptic neuron? The type of presynaptic neuron. The type of presynaptic neuron? Really? Oh, like whether it's myelinated or not? No, that's not. Well, that's true, right? In a chemical messenger, there's a chemical synapse. Then if It needs to be specific to the receptor. Oh, okay. Okay, sure. So we know from the other one, ionotropic was the ions diffuse directly into the postsynaptic neuron. Or right, was it postsynaptic neuron? Is that right? What do they call them? Is that right? Postsynaptic neuron? Yeah, postsynaptic neuron. And then they move. Well, they move kind of fast. Voltage sensing. I think it was one is was it the metabotropic? It's more widespread. Synaptic location. Oh, I think so, right? Effective activation. What? The other question said it didn't bind the ligand. Oh, I guess it was still. Let me write this down. We can review this a little bit. I must have forgot. Maybe it's on this video. Neurotransmitter. Compensate. It would be diffusion, from what I remember. Monoamines, I think. Okay, well, that's okay. I should be doing it faster, huh? We got 5, 5, so 5, 3, 3 and 5. Should take me like 8. Okay. Oh, geez, seriously? Well, oh, there we go. Oh, it's good a minute. So she decreased calcium. She decreased calcium. She decreased calcium. What happens now? She decreased the calcium. I can't talk. Maybe, possibly, right? I think she won't get any. You get 9. So, 2, 8, 8. So it could be 2. Huh? What? What are we talking about here? What? Oh, well, I guess. Well, it's still going to conduct the extra potential. I would have thought you'd get calcium in the axon terminal. Oh, but you wouldn't necessarily get an increase of it. And decrease in the number of listeners. This is false for sure. It's gonna be C. This weird wording. Why is it? It should be none, right? It would just be none. What do you mean none? Yeah. Okay. But well, why would I say none? No, decrease. Should be none. Sure. So in MS, that's definitely propagation. Or maybe B. Potentially A. B. Well, I did fire. That's so weird. Decrease neurotransmitter. Huh? Oh my goodness. Oh, I was completely wrong here. What the heck? Huh? All of them? Lack of neurotransmitters and the lack of synaptic vesicles in the axon terminal. A lack of synaptic vesicles does not suggest a defect in the potential propagation postsynaptic neuron. Oh, anterograde transport. Transports vesicles from the cell body to the axon terminal. You got me. You got me. Ugh. Ugh, that one took a hit. That one took quite a hit. That's okay. That's okay. That was a pretty good question. That was a good question. Sure. Let's just see how long these videos are. Ooh. Not bad. That's fine. Actually, I wanted to quickly see. Oh, not this one. Huh. Huh. What the heck? Oh, let's just quickly watch this one.
More accurately, it's the synapse that's excitatory or inhibiting. And even more specifically, it's the combination of the neurotransmitter that's released at the synapse. In this video, I want to talk about the types of neurotransmitter receptors. Neurons are often referred to as excitatory or inhibitory, but more accurately, it's the synapse that's excitatory or inhibitory. And even more specifically, it's the combination of the neurotransmitter that's released at the synapse and the receptor that it binds to on the postsynaptic membrane, because many neurotransmitters can bind to multiple types of receptors, so that the neurotransmitter can sometimes be excitatory and it can sometimes be inhibitory to the target cell. So that this axon terminal might be releasing this neurotransmitter here at the synapse, and perhaps when it binds to this purple receptor, that causes an excitatory potential in the target cell. But if it binds to this orange receptor on the postsynaptic membrane, that would cause inhibition of the target cell, inhibitory potential. When the target cell is another neuron, excitatory or inhibitory synapses can be scattered around all over the surface of the neuron. Or there are many neurons where the dendrites are receiving predominantly excitatory synapses, so that there can be a large number of neurons synapsing on the dendrites and releasing neurotransmitters that will cause depolarization in the dendrites. So let me just draw little purple pluses in here to represent that these are excitatory synapses. And then many neurons like this will have more inhibitory synapses on the soma. So I'll just draw some little orange minus signs here to represent that these could be inhibitory synapses on the soma. And then often neurons have synapses on their axon terminals, so that other axon terminals are synapsing on the axon terminal of the target neuron. And these will often be a mix of excitatory and inhibitory synapses. And there's a big variety in how the synapses are set up on neurons. And there would be a mix of some excitatory dendrites that can block those depolarizations from reaching the trigger, increase or decrease multiple level information flowing in. But there are some other big differences in neurotransmitter receptors and how they pass information from neurotransmitters in the chemical synapse to the target cell. So let me just draw a big axon terminal here that's releasing neurotransmitter into the synaptic cleft. And this will be the postsynaptic membrane of the target cell. And I'm going to draw the two big types of neurotransmitter receptors here. Let me draw this one in gray. It's a little receptor to bind to the neurotransmitter. And I'll draw this other one in yellow over here, with its little pocket to bind to its neurotransmitter. So this first type of neurotransmitter receptor is called ionotropic. And ionotropic neurotransmitter receptors are ligand-gated ion channels. So they have ion right in the name. And when their ligand binds to the receptor, which in this case is their neurotransmitter, they open and allow certain ions to pass. And when the neurotransmitter leaves the receptor, then they close and they don't let ions pass through anymore. The ionotropic neurotransmitter receptors cause graded potentials when they are activated, which have a rapid effect on the potential of the nearby membrane that is brief and local. The target cell will usually be excited if the activated ionotropic neurotransmitter receptor allows sodium or calcium ions to pass, because they will usually flow into the neuron, bringing their positive charges in and causing depolarization. Or the ionotropic neurotransmitter receptor will usually cause inhibition of the target cell if it allows chloride or potassium ions to pass. Chloride ions will usually flow into the neuron, bringing negative charges in, making it more negative inside. And potassium ions will usually flow out of the neuron, carrying their positive charges outside and also making it more negative inside.
Wait, what?
Now the other and more negative inside, and potassium ions or potassium ions to pass. Chloride ions will usually flow into the neuron, bringing negative charges in, making it more negative inside. And potassium ions will usually flow out of the neuron, carrying their positive charges outside and also making it more negative inside.
Get out
Now,
of here.
the other big class of neurotransmitter receptors are called metabotropic. And these are not ion channels. Instead, when their neurotransmitter binds to the receptor, they activate second messengers inside the neuron. And there are a number of different types of second messenger systems that can be activated by metabotropic neurotransmitter receptors. And these second messengers can do a lot of different things. They can go and affect the behavior of ion channels, causing them to be more or less active, or they can change the activity of proteins inside the neuron. And some can even affect the activity of genes and change the pattern of gene expression inside of neurons. When these metabotropic neurotransmitter receptors are activated, the response of the target cell occurs more slowly than it does with activation of ionotropic neurotransmitter receptors. But the effects may be larger and they may be more widespread because there can be amplification through these second messenger systems. The effects of activation of these second messenger systems by metabotropic receptors may involve just brief excitation or inhibition of the target cell, or it may cause longer lasting changes to how the target cell behaves, either when it's at rest or when it's responding to input.
All right. I guess that's what I messed up on, huh? Ionotropic, ion-gated, or ligand-gated ion channels, ionotropic, allow certain ions to pass through. But if they're ions, then they're already going to be smaller, right? But if they pass through, I mean, it also depends on the structure, right? Because the cell membrane itself is semi-permeable and hydrophobic. So it depends. I would think it depends. But they are relatively small. Whatever. The question's gone. Let's stop worrying about it. Yeah. Okay. Highly recommend. Highly recommend you do one Jack Weston free passage a day. Okay. Thanks, Adine. I think I read on Reddit that he had made the books free or something. I keep hearing about this Jack Weston guy on Reddit like all the time. People love him. He's like the Messiah or something. I don't know. But maybe I'll try to look for it. Let's see. Jack Weston Forest passages. Is it like somewhere here? I don't know. How do you work this? Oh, there's today's daily passages, and it just goes back in time. Wow, how far does it go back? Oh, what happened? What happened? Oh, okay. Wait. Go back? What happened? Section cars? I don't know. How do I work the site? Maybe. What am I looking at? Question maybe? Sections. Nope. I don't think so. I don't know. I just leave it here, I guess. But how can I? There's no next. It doesn't work. card. No, I don't want to do no enrolling. Self-study. Casper shortly. I don't know. I mean, I think let's not spend too much time on this. I think maybe just these are the only ones I can do, right? What am I doing wrong? Why can't I go back? It's really bad. I'm like, is it just me? Should I switch to Google or something? I have Internet Explorer. I use Internet Explorer for some of my stuff. Well, most of my stuff. Because I can't go out of it. I mean, this is already a good set right here, right? 1, 2, and then 1, 2, 3. 15, 17 here. Yeah, the website's just bad. Oh, okay. That's okay. Well, there's 17 here, so I guess that's 17 days. Maybe that's still not bad, I think. Each day there's a new passage. So maybe just leave it here, right? Because that way, like maybe, so each day there's a new passage, today's religion. And I can hit some of these, I guess. So I'm guessing like tomorrow then it'll be a new passage that'll be here. Is that what I'm thinking? Yeah, well, what happened to religion? It's already gone? Oh, no, it's still there. Okay. Yeah, thanks so much, Nadine. I appreciate it. Okay, yeah, I will. I'll do one card section. Yeah, thank you. Maybe we can do it tonight or something. I think I might have to. We finished Anki today. I'll add the cards tonight, and then we can do one. Oh, well, yeah, thank you. I appreciate it. Any help I get, I appreciate it. Okay, yeah. Around the right. I think it should be 3.5 minutes to read, 30 seconds to write a main idea, and a minute per question. Something like that. Oh, yeah, my timing is bad. I'm gonna take my first full length. Well, maybe Sunday or this Monday, something like that. Oh yeah, where we at? Okay, let's review a little bit. I wanted to review some gaps. Thanks. Yeah, I had some gaps on the what was it? I don't know. Well, we can just go through some of the gaps. It's fine. I'll just share this too. Whatever. Who cares? There was someone that posted on Reddit like a prompt for ChatGPT where it's like if you have any questions or something, it's gonna break it down real nice. So I grabbed this whole post, to be honest, and then I just put it on ChatGPT. So now when I ask a question, it's set up in the optimized way or the way that like was more optimal or something to have like a study buddy or something. Because I mean, I don't have the money to pay for a tutor, and this is free. So, but anyways, let's just take like a 10-minute break. So we'll take a 10-minute. We'll finish this review a little bit, and then we'll get started. Endocrine and circulatory today, right? So that's what we got here today. I guess I'll just leave it here. I'll be back. Get inside. Take your nap. Good night. I don't know. I'm not here, Mom. Oh man, my mother's so funny. Oh. Yeah. I want to eat, but I'm trying to lose some. I'm trying to lose a little bit of fat, so maybe I should. I already had the ice cream too. That's already too much. Okay, let's do it. Mm-hmm. Oh, I think the connection's bad, huh? Can you guys see? Okay, flipping a coin, rolling a die. Actually, let's put this over here. Oh, yes, I know. As long as I have the clock. So, two hits in a row. Just multiple time to each other. Independent flip doesn't affect the other. Independent and multiply. Okay. Mm-hmm. They both can happen at the same time. Oh, you can't get an A or an A. You can only get one? One at a time. Mm-hmm. Mm-hmm. Right. A, B. Yeah, there you go, something like this. What is the chance of getting AA and BB from two separate genes? Two independent events. Two events are independent of one affect the other. One happening does not affect the other. One happening does not affect the other. Okay, what's the chance again? So what's the chance of getting AA and BB? Which is AAPB? But I think so. You know to take it because A B B B B. Is how it's written, right? How the genotype is written? I think so. Let's try. I really don't want it to give me the. I want to try some out. I mean, does it matter? Isn't it always going to be like this? Look like this? A B 4 to 16. A both heterozygous. Both heterozygous. So both heterozygous. 1, 2, 3, 4. 4 to 16. Homozygous. 1, Okay, 2 out of 16. How much is that? Okay. Yeah. It's like laggy, so we have to wait. I think I have like 3 of these. I think the other ones are not too bad. I just want to practice some of the button squares. I don't know why. Okay, let's see. A basic one. I mean, the basic one is just 1, 4, right? And then I think 1, 2, AA or AA. So we know that AA is 2 out of 4 plus A. What? From the same AA cross. Two children in a row. 2 children in a row, right? Two children in a row. So well, it's 1/4. So that turn to 1/16. And that's going to be the same for BP. I don't know, BP. With AA and so AA and BP. Is that it? AA BP. So for 5, is that saying it's going to be 1/2 times 1/2 plus 1/4 times 1/2? So 1/4 plus 3/8. I think for 2, no, 2's fine, I think. Let's see. 1/4. 2. 2. Is that it, right? Isn't it the same? Still 1/2, right? This is fine, then I think I'm okay. And we'll hit the other ones, and then we will it's like 10 minutes each, right? Well, less than that, to be honest. I think we'll just maybe we can exercise a little bit. If this is all correct, then I think we'll take a short break, just exercise real quick, and then we'll go ahead and start lesson 5. Oh, we did. Okay, yeah. I mean, let's. Yeah, that's very good. That makes me happy. Makes me happy. Makes me happy. I understand it. Okay. Good. Cool. Very nice. Where am I at now? Okay. Yeah. Let's see. I'm on this one. Cool. Okay. So first, the electrochemical gradient, which combines both concentration gradients and electrical gradients. Excuse me. Mm-hmm. These particles move in the same direction. Yeah, I understand. No, I got that. I meant, yeah, but which one in particular? Was there anything else? I think it was just those two, right? Oh, I lied. Yeah, just wait for it. Oh. 579. Gotcha. Okay, that's it. That's what I wanted to know. That's all I wanted to know, buddy. Thank you. Anything else? Do you have any other questions? Any other questions? Any other questions? I think that's all stuff from before. Okay, now we're good. Okay, cool. Now we're okay. Let's go back now. I think, well, yeah, that was what I had. So that's it. That's all I wanted to review real quick. Good. Very good. Very good. Let me go ahead and make a shake real quick. Not a fruit shake, really. Just a powder shake. And nice. It's already 16. We'll do these two. Well, when I come back, we'll exercise real quick, and then we will finish these two and do a cars passage. All right. I mean, sorry guys, but anyways, I think the best thing right now is okay. Let's let's at least finish this one first, right, before we begin to work out. I don't want to. It's already 5, so we need to hit it. Let's go.
Have you ever thought about the way
Whoa, sorry.
parts of our body communicate? I think we often consider the body to be this one complete thing, this self. But really, our body is composed of lots of parts. There are lots of organ systems, and each of those has organs, and all of those organs are made of tissues, and all of those tissues are made of cells.
Oh, let me just answer. Okay. All right. Let's go.
And it's crazy that there are 100 trillion, or at least roughly 100 trillion cells in our body. So it's curious then, how do those 100 trillion different parts communicate? Well, one way is through the nervous system and through the prelate tracts of nerves. But not every part of the body is connected by nerves. I mean, how, for example, would part of the brain go about communicating with part of the kidney? Well, to talk about that, roughly, and all of those organs are made of tissues, and all of those tissues are made of cells. And it's crazy that there are 100 trillion, or at least roughly 100 trillion cells in our body. So it's curious then, how do those 100 trillion different parts communicate? Well, one way is through the nervous system and through the prelate tracts of nerves, but not every part of the body is connected by nerves. I mean, how, for example, would part of the brain go about communicating with part of the kidney? Well, to talk about that, we're going to have to talk about the endocrine system. And the endocrine system is a system of organs that are called glands. It's curious then, how do those 100 trillion different parts communicate? Well, one way is through the nervous system and through the prelate tracts of nerves, but not every part of the body is connected by nerves. I mean, how, for example, would part of the brain go about communicating with part of the kidney? Well, to talk about that, we're going to have to talk about the endocrine system. And the endocrine system is a system of organs that are called glands. And these glands secrete little chemical messages that are called hormones. And they release those little chemical messages called hormones into the bloodstream so that they can circulate from one part of the system. And these glands secrete little chemical messages that are called hormones And the endocrine system is a system of organs that are called glands. And these glands secrete little chemical messages that are called hormones. And they release those little chemical messages called hormones into the bloodstream so that they can circulate from one part of the body to another part of the body in order to initiate an effect. And there are many parts of the body that use these hormones to communicate, but certain organs are really defined by this method of communication, and we call them endocrine glands. And so one of the major endocrine glands is the hypothalamus. It uses these hormones to communicate, but certain organs are really defined by this method of communication, and we
So fascinating, isn't it?
call them endocrine glands. And so one of the major endocrine glands is the hypothalamus.
The hypothalamus.
And the hypothalamus is located right here. It's a member of the forebrain, and as a member of
Yes, sir.
the
It's a member of the forebrain.
brain, it receives a lot of those signals that we talked about from the nervous system. So those nerve signals are funneling into the brain, and the hypothalamus then, as a kind of dual member of the endocrine system, funnels those signals into the pituitary gland. And so because it plays that dual role between the endocrine system and the nervous system, it often gets taglined as the control center of the endocrine system. In addition to stimulating the pituitary gland, the hypothalamus actually makes some hormones itself also. And so it makes ADH.
Here we go. Chemicals. I'm just a chemical. I'm just a chemical.
And oxytocin. And
So we're all just chemicals.
ADH is antidiuretic hormone, and it's
Antidiuretic.
a main regulator
Why did it get louder?
of our fluid. And ADH is antidiuretic hormone, and it's a main regulator of our fluid volume in our body. And then oxytocin is a hormone that stimulates the uterus to
Oxytocin stimulates the uterus.
fluid volume in our body. And then oxytocin is a hormone that stimulates the uterus to contract for females during pregnancy.
Oh, okay. Interesting.
And so that's the hypothalamus, member of the brain and member of the endocrine system, where it all begins, the control center. And then right below the hypothalamus is the pituitary gland. The pituitary gland is located right here, dangling right below. And so the hypothalamus is about the size of a grape. And the pituitary gland is actually about the size of a green pea. But this little green pea is so important that it's called the master gland.
The master gland.
And it's called the master gland because the pituitary gland takes that stimulation from the hypothalamus and it directs it to all of the other endocrine glands. Body that use these hormones to communicate, but certain organs are really defined by this method of communication. Endocrinists talk about the brain effects of nerves, but not every part of the body is connected by nerves. I mean, how, for example, would part of the brain go about communicating with part of the kidney? Well, to talk about that, we're going to have to talk about the endocrine system. The messages that are called hormones. And there are many parts of the body that use these hormones to communicate, but certain organs are really defined by this method of communication, and we call them endocrine glands. And so one of the major endocrine glands is the hypothalamus. And the hypothalamus is located right here. It's a member of the forebrain, and as a member of the brain, it receives a lot of those signals that we talked about from the nervous system. So those nerve signals are funneling into the brain, and the hypothalamus then, as a kind of dual member of the endocrine system, funnels those signals into the pituitary gland. And so because it plays that dual role between the endocrine system and the nervous system, it often gets taglined as the control center of the endocrine system. In addition to stimulating the pituitary gland, the hypothalamus actually makes some hormones itself also, and so it makes ADH and oxytocin. And ADH is antidiuretic hormone, and it's a main regulator of our fluid volume in our body. And then oxytocin is a hormone that stimulates the uterus to contract for females during pregnancy. And so that's the hypothalamus, member of the brain and member of the endocrine system, where it all begins, the control center. And then right below the hypothalamus is the pituitary gland. And the pituitary gland is located right here, dangling right below. And so the hypothalamus is about the size of a grape. And the pituitary gland is actually about the size of a green pea. But this little green pea is so important that it's called the master gland. And it's called the master gland because the pituitary gland takes that stimulation from the hypothalamus and it directs it to all the other endocrine glands, or at least almost all of the other endocrine glands, such that their function is ultimately dependent on the pituitary gland to work well. And so that little green pea is a really important part of the endocrine system. And so one of the endocrine glands that the pituitary directs is the thyroid gland. And the thyroid gland is located right here in your neck. It wraps around your trachea. And the thyroid gland is located right here in your neck. It wraps around your trachea. And your trachea is your windpipe.
It's in the neck. It's in the neck. What the heck? Why is it in my neck?
And so you can feel this thyroid. And your trachea is your windpipe. And so you can feel this thyroid gland on your neck as you swallow. If you hold your hands right around your Adam's apple and swallow, that meaty thing moving up and down, that's your thyroid gland. And one of it,
I don't know. I don't feel anything. I have to look it up now. Thyroid gland. The meaty thing. Thyroid gland. Oh, what the heck? That's a neck mass. Where is it? Oh, it's here then. Oh, it's right here. Oh, okay. Whatever. Whatever, it's somewhere there. Okay, I got it. I understand.
Its main jobs is regulating your body's metabolism. Right here in your neck, it wraps around your trachea. And your trachea is your windpipe, and so you can feel this thyroid gland on your neck as you swallow. If you hold your hands right around your Adam's apple and swallow, that meaty thing moving up and down, that's your thyroid gland. And one of its main jobs is regulating your body's metabolism. And it does that through the thyroid hormones T3 and T4. And another name for T3 is triiodothyronine, and another name for T4 is thyroxine. But the thyroid uses these hormones, the thyroid hormones, to stimulate the body's metabolism, which is crucial because that's how our body gets energy. And then right behind the thyroid gland are 4 spots known collectively as the parathyroid. And the main role of the parathyroid is regulating our body's blood calcium level. And the level of calcium in our blood is hugely important because calcium does a lot of stuff in our bodies. It's involved in muscle contraction, it's involved in bone growth, and all of those functions are really sensitive to the level of calcium that's floating around in our blood. And so the parathyroid glands, those 4 spots on the backside of our thyroid, regulate calcium through the hormone, parathyroid hormone, or PTH. And then moving down the torso, we have the adrenal glands. And the adrenal glands are located right on top of the kidneys here. And they're called the adrenal glands because they're adjacent to or right next to the kidney system, which is called the renal system. In medical speak. But we really need to further divide the adrenal glands into 2 parts, the outer part and the inner part. So the outer part is the cortex. And the inner part is the medulla. And the reason for the distinction is that the inside and the outside of the adrenal glands have 2 different functions. And so we'll start with the outside, or the cortex, and that's where the steroids, the adrenal corticosteroids, are made. And 2 major examples of steroids made in the adrenal cortex are cortisol and aldosterone. And cortisol is one of the body's stress hormones, so it functions to increase blood sugar in times of stress, so we have energy, and it also has some anti-inflammatory functioning. And then aldosterone is one of the major regulating hormones of our body's blood volume and how much fluid is in our veins and arteries. And so that's the cortex. And then the medulla makes a class of hormones called catecholamines. And 2 major examples of catecholamines are epinephrine and norepinephrine. And I'm going to shorten those as epi and norepi. And sometimes epinephrine is called adrenaline, and that might be a little bit more familiar to you, but these catecholamines are really involved in our body's fight or flight response, that adrenaline response that we have to a stressful or scary situation. And so the medulla and the cortex make up the adrenal glands, but moving down the list and down the body, we have the gonads. And in females, those are the ovaries. And in males, the testes. And the gonads release the sex hormones. And so in males, the testes produce testosterone, and in females, the ovaries produce estrogen and progesterone. But these sex hormones are mainly involved in the development of our secondary sex characteristics like pubic hair and larger frames in males and breasts in women. But they're also involved in progressing us through those life stages that accompany those sex characteristics like puberty and menopause. And then last but not least, we have the pancreas. And it's located right here in the upper part of the abdomen. And I saved the pancreas for last because it isn't involved as directly with the pituitary glands as the other endocrine hormones were, but it still uses those hormones to stimulate an effect in a different part of the body. And the effect that the pancreas stimulates is control over the blood sugar. And it does that through the hormones insulin and glucagon. And the pancreas is vitally important because without its hormones insulin and glucagon, we can't regulate how much sugar is in the body's blood versus the cells. And that can lead to major diseases like diabetes, the blood sugar. And it does that through the hormones insulin and glucagon. And the pancreas is vitally important because without its hormones insulin and glucagon, we can't regulate how much sugar
It'd be
is in the
nice
body's
if we finish
blood
this early.
versus the
Let's take a
cells.
cold
And
shower.
that can lead to major diseases like diabetes. And
Well,
so
maybe we
with
should
the
do it
pancreas,
after.
we can conclude our list of major
We should do it
endocrine.
after.
And so as we look at these glands and at these hormones, and we think about all of the different effects that are being stimulated in our body by them, it becomes pretty clear that there aren't just a few of these circulating in our bloodstream. There are literally loads of hormones circulating through our vasculature at any given moment. And so that poses a potential problem. If, say, that you're in the brain and you're trying to tell something to the kidney, you're trying to send them a message, and you put that in the bloodstream and you just float it down to them, how do you know that it's going to get there? I mean, isn't that what every other endocrine gland is trying to do? Well, it turns out that hormones are a lot like radio waves. In your city or in your town, there are many different radio stations, and there are many different songs being played at any given time by those radio stations. And even maybe from the next town over, there are radio waves filling the air of your town. But unless you're tuned in specifically to that station, you're not going to pick up on the song that's being transmitted. And in a very similar way, a hormone is not going to be received unless there's a very specific receptor on the target cell. And so the receptor and its location are very important in determining the hormone function. And we have classes that we use to help us identify which hormones fall into which function. And so the first class are autocrine hormones. And so the receptor and its location are very important in determining the hormone function. And we have classes that we use to help us identify which hormones fall into which function. And so the first class are autocrine hormones.
Autocrine hormone.
Hormones
Why is it playing that?
function at the cell that makes them. An example of this is the T cell in the immune system. system. It actually secretes a hormone that it makes called an interleukin that signals the cell itself to increase its effectiveness in its immune function. And then another class of hormones are paracrine hormones. And paracrine hormones function regionally. And an example of that might be the hormones released by the hypothalamus that direct the pituitary gland. And then last but not least, kind of the class that direct, an example of that might be the hormones released by the hypothalamus that direct the pituitary. And then last but not least, hormones are the endocrine hormones. And these are the and an example of this might be the pituitary gland stimulating the gonads. Way far away. And so we have autocrine, paracrine, and endocrine classes that help us determine how a hormone functions. And so I know I just told you a whole lot about hormones, but this is your introduction into one of the most important ways that the 100 trillion little tiny individual parts of your body communicate.
Okay. Okay, so today what I want to talk about is endocrine control. And in order to talk about endocrine control, I need to talk about two major glands. First, the hypothalamus. I'm going to draw that in here. Then in this enlarged image right here, this is just a blown up view of the hypothalamus. And then the next major gland that we need is the pituitary gland. And the pituitary gland is the gland that dangles right below the hypothalamus. And you can see that the hypothalamus is a structure right here in the forebrain, and the pituitary dangles right beneath it. And as a member of the brain, the hypothalamus receives neural signals from the brain and from the peripheral nervous system, and it funnels those signals to the pituitary gland, which ultimately controls the other endocrine glands and our body's hormonal response to the environment. And there are two different parts to the pituitary gland. You have the anterior pituitary gland, and then you have the posterior pituitary gland. And the hypothalamus interacts with the anterior and posterior part in two different ways. And so it interacts with the anterior pituitary gland primarily through the hypophyseal portal system, which I've kind of drawn in here. And the hypophyseal portal system is you have the anterior pituitary gland.
And then you have the posterior pituitary gland. And the hypothalamus interacts with the anterior and posterior part in two different ways. And so it interacts with the anterior pituitary gland primarily through the hypophyseal portal system, which I've kind of drawn in here. And the hypophyseal portal system is a capillary system, so little blood vessels that flow between the hypothalamus and the pituitary gland, and the hypothalamus secretes hormones into this little system, and they go down and they signal the pituitary gland. And so that would be an example of a paracrine signal, or a really regionally active signal. And so one example of the hypothalamus hormones that signal the pituitary gland is gonadotropin releasing hormone, or GnRH. And gonadotropin releasing hormone is going to go down to the anterior pituitary, and it's going to stimulate the release of follicle stimulating hormone and luteinizing hormone, so FSH and LH. And these hormones are going to travel down to the gonads in the male to test L hormone and luteinizing hormone, so FSH and LH. And these hormones are going to travel down to the gonads in the male, the testes, and the female, the ovaries, and they're going to stimulate the gonads to release their hormones. And that's in the female, the ovaries, and they're going to stimulate the gonads to release their hormones. Another example of how the hypothalamus controls the pituitary gland, which ultimately controls the endocrine glands, is corticotropin releasing hormone. And corticotropin releasing hormone stimulates the anterior pituitary's release of adrenocorticotropic hormone, ACTH. Adrenocorticotropic hormone goes down to the adrenal glands, and it stimulates the adrenal glands release of its hormones. And the adrenal glands, and it stimulates the adrenal glands release of its hormones. And so moving along, the hypothalamus also releases thyroid releasing hormone, TRH. And thyroid releasing hormone goes down to the anterior pituitary, and it stimulates the anterior pituitary's release of thyroid stimulating hormone, TSH. And thyroid stimulating hormone goes down and it stimulates the thyroid gland to release its hormones, thyroxine and triiodothyronine. And so, continuing in that list, you have growth hormone releasing hormone. And growth hormone releasing hormone is the hypothalamus' signal to the pituitary gland to release its hormone, growth hormone. And growth hormone goes to the long bones and the big muscles in our body and stimulates growth. And then last but not least, we have prolactin inhibitory factor, PIF. And prolactin inhibitory factor is a little bit different because it's constantly being released, and when it stops being released, that's when the pituitary gland is signaled to release prolactin. And prolactin is a hormone involved in milk production in moms. And so some of the anterior pituitary hormones go down and directly stimulate other endocrine glands like FSH and LH. but some of the anterior pituitary glands directly affect parts of the body, like growth hormone and prolactin. And so there's a nice mnemonic, flat peg, which helps me remember which one's which, because FSH, LH, ACTH, and TSH, or the flat hormones,
What?
T8, that indirectly stimulate other endocrine glands like FSH and LH. But some of the anterior pituitary glands directly affect parts of the body, like growth hormone and prolactin. And so there's a nice mnemonic, flat peg, which helps me remember which one's which, because FSH, LH, ACTH, and TSH, or the flat hormones, are called tropic hormones. Let me get a different color for this. And the tropic hormones are hormones that stimulate other endocrine glands. PEG hormones are the direct hormones, and they include prolactin and growth hormone, and the E is for endorphins, which I haven't included in this list because the anterior pituitary does release endorphins, and endorphins are hormones. The LH, which helps me remember which
Hey, hold on, guys. I have to move the car. I'll be back. I said I think that leaves the in and out of the table. We just got some in and out. You know? Just got some in and outy. Food is food. You gotta eat. Hold on. Thick thighs. They say thick thighs save lives. You guys heard that one? They say thick thighs save lives. Thick thighs save lives. Oh my goodness. I almost can't say it. Thick thighs save lives. All right. Doctor's here. Doctor's here. I'm messing around. Okay, never mind. I'm not gonna say it. Okay, anyways, let's keep going. Let's maybe watch the video, finish watching it, watch it twice, and then as we just watch the other videos. Right? As I said, I was gonna say a joke. I was gonna say a joke, but it might not be that funny, so it's okay. We'll finish this and I want to replay the video, to be honest.
SH, LH, ACTH, and TSH, or the flat hormones, are called tropic hormones. Let me get a different color for this. And the tropic hormones are hormones that stimulate other endocrine glands. PEG hormones are the direct hormones, and they include prolactin and growth hormone, and the E is for endorphins, which I haven't included in this list because the anterior pituitary does release endorphins, are called tropic hormones. Let me get a different color for this. And the tropic hormones are hormones that stimulate other endocrine glands. PEG hormones are the direct hormones, and they include prolactin and growth hormone, and the E is for endorphins, which I haven't included in this list because the anterior pituitary does release endorphins, but so do a lot of other parts of the body. And so E is for endorphins, but the PEG hormones are direct hormones. And direct hormones stimulate a part of the body directly. So growth hormone directly stimulates the bones and the muscles, and prolactin directly stimulates lactation. And so the hypothalamus signals the anterior pituitary's release of its hormones to the hypophyseal portal system, or this little capillary bed. And then the hypothalamus also communicates with the posterior pituitary, and it does that through stimulation of nerves which run down that pituitary stalk right here. And the hypothalamus sends a signal down those nerves to the posterior pituitary and causes the posterior pituitary to release a couple hormones too. And I want to make a point because the posterior pituitary releases these hormones, but they're actually made in the hypothalamus and stored in the posterior pituitary. And the two major hormones are ADH and oxytocin. And ADH is antidiuretic hormone, and it stimulates the collecting ducts in the kidneys to retain water. And oxytocin is a hormone involved in uterine contractions in women. And so you see that a lot of the endocrine control in the body ultimately comes back to the pituitary gland, which is then controlled by the hypothalamus, which forms the bridge between the nervous system and the endocrine system. And so as big as the names of the hormones get, the idea behind them is pretty manageable. It's really just one part of the body stimulating another part of the body through chemical messages.
Right. Okay. I'm gonna say it. I'm gonna say it. In-N-Out is the best burger place. In-N-Out is the best burger place. At least